Hello everybody, I am Dr. Dolly Nigam and today I am going to talk about cancer, the most dreaded disease of all time. So let's see what cancer is and how it is developed, what are the causes of the cancer. We'll see about it. So cancer is a fundamental abnormality of the cells. In this, the cells continually proliferate in unregulated and uncontrolled manner rather than responding appropriately to the signals that control normal cell behavior or normal cell division. Cancer cells grow and divide in an uncontrolled manner, invading normal tissues and organs and eventually spreading throughout the body. So, this uncontrolled growth of cancer cells is the net results of all accumulated abnormalities in multiple cell regulatory system. There are n number of cell regulatory systems during the translation, transcriptions, DNA replications and cell divisions different at different stages, G0 stage, G1 stage, synthesis stage, prophase, metaphase, anaphase. So all those there are checkpoints when that decides and determines when the cell has to enter to the cell division phase. So all those checkpoints are disrupted or started malfunctioning in the cancer cells. So that's why cancer cells behave differently and their normal counterparts. So let's see what are different types of cancer. So cancer can occur in any of the different types of cells that are present in our body. So there are more than 100 distinct types of cancer. The most important issue in cancer pathology is the distinction between benign and malignant tumors. So just let's see what is benign and malignant tumors. So benign benign literally means gentle and kind. So a benign tumors remains confined to its original location. Just think about what tumor is. Tumor literally means it's a swelling of any kind. So a tumor is any abnormal proliferation of cells which may either be benign or malignant. Benign means gentle or kind. So these cells, these proliferating cells remain confined to its original locations. So they do not move from one side of the body to another side of the body, and that is why they are just localized. They have some abnormalities, but those just at one particular time they stopped cell dividing. Okay, so here the proliferations is localized at one particular site. It neither invades surrounding normal tissues nor spreads to distant body sites. Example, skin warts. Suppose you have seen some people have skin warts, little swelling, black color swellings or even white color swellings. So these are the examples of benign tumors. Now let's see what is malignant tumors. So malignant literally means evil. So malignant, evil in nature or evil in effect. So these malignant tumor is capable of invading surrounding normal tissues and spread throughout the body via the circulatory or the lymphatic systems, because either they can circulate through the blood or either they can circulate through the lymph. Lymph is another body fluid that are present in our tissues, all the fluid that are present in the tissues and intracellular spaces. So these are the limbs, and all through the limbs or through the blood they can reach anywhere and cause and start proliferating then and there. So this condition, suppose this invading surrounding, this capability of cancer cells of invading surrounding normal tissues or neighboring tissues and spread throughout the body, this particular condition is called metastasis. So only malignant tumors are properly referred to as cancers, and it is their ability to invade and metastasize that makes cancer so dangerous. Whereas benign tumor can be removed surgically, and the spread of malignant tumors to different body sites frequently makes them resistant to such localized treatment, because once suppose you have surgically removed that malignant tumor, and if it has got already into the metastasis stage, so this particular cell, even a single cell, can reach to another cell and there they can start proliferating. So that is why malignant tumor is very difficult to treat. So both benign and malignant tumors are classified according to the types of cells from which they arise. Most cancers fall into one of the three main groups. What are these three main groups? One is carcinoma, the other one is sarcoma, and the third one is leukemia or lymphomas. So let's see what carcinoma is. So carcinoma includes approximately 90% of human cancers. These carcinomas spread in the epithelial cells. Then sarcoma are rare in humans. They are the solid tumors of connective tissues such as muscles, bones, cartilage and fibrous tissue. Then leukemia or the white blood cells. So leukemia and lymphoma account for approximately 8% of human malignancies. Leukemia arise from blood forming cells. These are the hematopoietic cells. All the blood cells, whatever bloods, RBCs, WBCs, they are formed and produced in the bone marrow. So these from bone marrow, and they are arising from the stem cells that are present in bone marrow. So these stem cells are called hematopoietic stem cells. It means that those stem cells that are giving rise to different kind of blood cells. So leukemia cancer arise in the blood forming cells, and lymphomas are from the cells of the immune system, like different mast cells or tissue cells. So although there are many kinds of cells, only a few occur frequently. Cancers of 10 different body sites account for more than 75% of this total cancer incidence. So the four most common cancers accounting for more than half of the cancer cases. Let's see what are those four cancers that account for more than half of the incidences. So these are breast cancer, prostate cancer, lung cancer and colon or rectum cancer. The lung cancer by far is the most lethal one, and it is responsible for nearly 30% of all cancer deaths. So let's see how this cancer develops in our body. So the development is a multi-step process in which cells gradually become malignant through a progressive series of alterations or mutations. Suppose one mutation is occurring, then the body different repair mechanisms is also acting, but continuously these are accumulated, these alterations, one mutation, second mutation, third mutations, n number of mutations are accumulated, and it will give rise to certain kind of malignancies that will lead to cancer. So one indication of the multi-step development of cancer is that we see that most cancer develop late in life, barring few, few, and the incidence of the colon cancer, for example, increases more than tenfold between the ages of 30 and 50, and another tenfold between 50 and 70. So such a dramatic increase of cancer incidence with age suggests that most cancers develop as a consequence of multiple abnormalities which accumulate over periods of many years. So generally we see कि when we age, and in older people or old age people, many cancers are developed. So at the cellular level, the development of cancer is a multi-step process involving mutation and selection for cells with progressively increasing capacity for proliferation. They want to grow and proliferate. They don't care what are the other cells, what are the normal cells are lying, so we don't have to grow. But here what happens, they just have different kind, they just solely, they are destined to grow, rapid growth, rapid growth. This is the whole and soul nature, whole and soul characteristics of the cancer cells. So first, just see how this cancer developed in different kind of stages. The first step in this process is tumor initiation. We must know that tumor is initiated by just a single cell, abnormality in a single cell. So it is the result of genetic alteration or mutation that leads to abnormal growth of a single cell. This abnormal cell proliferation leads to the outgrowth of population of tumor cells. The second stage is tumor progression, wherein additional mutations occur within the cells of tumor population. So these proliferative cells give rise to a small benign neoplasm, an adenoma or polyp. So what is the neoplasm? So neoplasm is basically the abnormal growth of tissues. Benign literally means, we all know that it means gentle or mild. So here the growth remains localized, such as in skin wall. So when some of these mutations, so at the same time it is arising from the single cell, and these single cells are rapidly with the accumulating some kind of mutations one by one. Descendant of these cells are also accumulating mutations. So after that, what happens? The neoplasm generates, the neoplasm forms, meaning abnormal tissue growth is formed, and that results in a simple kind of swelling in the skin, like such as in skin wart. So some of these mutations confer a selective advantage to the cell, such as more rapid growing cells are selected and become dominant within the tumor population. Suppose out of this, a teacher selects only a few students who is very good in handwriting, who is getting very good marks. So here what happens, the tumor cells, out of them, they select a particular cell which is showing more rapid growth, which is showing more survival, and which is showing more growth even without the lack of any growth factor requirement. So these particular clonal selection happen. It gives the selective advantage of the tumor population cells. So what happens? This particular selection is called the clonal selections. Since a new clone of tumor cells has evolved on the basis of its increased growth rate or other properties such as better survival, invasion, the capacity to metastasize, so that is conferring them a selective advantage. So this clonal selection continues throughout tumor development. So tumors continuously become more rapid growing and increasingly malignant. Remember that during progression, tumor progression, when the single cell is having any kind of abnormality and starts growing, it reaches the benign conditions. At that time it is not malignant, but after some times when so many other mutations is also occurring and one particular cell is showing certain kind of advantage of certain kind of rapid growth, better survival, then this particular clone is selected and they start growing. And then after these clonal selections, the malignancies can be started. So a single initially altered cell here gives rise to a proliferative cell population which progresses first to benign adenomas of increasing size and then to malignant carcinomas. The cancer cells invade the underlying connective tissues. So here, this way the cancer cells develop. So first, tumor is initiated by the abnormality or alterations or genetic mutations in a single cell. This single cell start growing without any inhibitions, they have certain characteristics, and then they reach into the tumor progression. After tumor progression, one particular clone is selected, that is clonal selection, and clonal selections after that, malignancies can be started. Okay, I hope this particular development of cancer is clear to you, all of you. And now come to the causes of cancer. So what are the different causes of cancer? What can lead to cancer in our body? So there are certain substances that can cause cancer. These substances are called carcinogens. So carcinogens have been identified both by studies in experimental animals and by epidemiological analysis of cancer frequencies. So there are many agents or potential carcinogens, but main are the three. So main are the three ones, that is UV radiation, chemicals and oncogenic viruses. So radiations and many chemical carcinogens, how they act? They act by damaging DNA and inducing mutations. So chemical carcinogens are basically the alkylating agents. These alkyl add a methyl group in the bases. So adenine and guanine bases, they add the methyl group, and because of this there is A, T and GC combination is disrupted. The A can bind to G or A can bind to C. So this kind of mutations occurs by the chemical carcinogens, and radiations also break down the DNA. It can break the chromosomes also. So these carcinogens are generally referred to as initiating agents. They are initiating agents. So since they induce mutations in genes that will lead to cancer development. Some of these initiating agents are solar ultraviolet radiations, that is the major cause of skin cancer. Carcinogenic chemicals generally are present in tobacco smoke, that is benzopyridine, and aflatoxin, that is a fungal toxins. This is a potent liver carcinogen. These are produced by some fungi mucor molds that are very toxic, and these aflatoxins create mutations in our body. So the other tobacco smoke actually contains so many different kind of chemical carcinogens, including benzopyridine, dimethyl nitrosamine and nickel compounds. So these are actually identified as the major causes of human cancer. And smoking is undisputed cause of 80 to 90% of lung cancer, as well as being implicated in the cancers of oral cavity, pharynx, larynx, esophagus and other sites also. So in total, it is estimated that smoking is responsible for nearly 1/3 of all cancer deaths. So it is an impressive toll for a single carcinogenic agent. So we should avoid smoking. Everybody, all students, any person, smoking is injurious to health. So just get rid of it. Anyways, now come to the other point. What are the properties of cancer cells? What are the characteristics of cancer cells? How can we distinguish that this is the cancer cell and this is not the cancer cell, this is the normal cells? So we all know right now, we have already understood that the uncontrolled growth of cancer cells result from the accumulated abnormalities. Let's see what aspects that cancer cells differ from their normal counterparts. So cancer cells typically display abnormalities in the mechanism that regulate normal cell proliferation, differentiation and survival. So first is there are different type of characteristics and we'll see one by one what are those characteristics. First is density dependent inhibition. So normal cells display density dependent inhibition of cell proliferation. So what is density dependent inhibition? So normal cells grow until they reach a finite cell density. They then stop proliferating and become quiescent, that is arrested in the G0 stage of GO or G0 stage of the cell cycle. This uncontrolled growth is partly determined by the availability of growth factors added to the culture medium. So before going to the this particular theory, we must know how these cells, the cancer cells, cell lines are provided for various experimental purposes. So there is a very interesting story behind it, and this was the story of Henrietta Lacks. Henrietta Lacks was the patient of cancer, of cervical cancer, and she died at the age of 31 while she was receiving her treatment at Johns Hopkins Institute in America. So while she was having while she was having those treatment, one of the cell, a small patch of tissue was taken out from a cell and given to Dr. Gay. And Dr. Gay used to have a device. So in that they used to make different type of cells, and they used to grow in different type of cell cultures. So generally what happens ki after 20 to 50 generations the cell eventually die. But what he see that in HeLa, that is in Henrietta Lacks cells, it keeps on growing and growing and growing. So it generally he collected it ki this a particular characteristics, particular different characteristics of this particular cells that keep on growing even on the petri dish, that even they make overlay one layer. Because normal cells, what happens, normal cells grow in one layer, only one layer. But what happens, this Henrietta Lacks cells keep growing and growing in the layers and layers and layers and layers. So that is why this HeLa cell lines have been discovered. And at that time Henrietta Lacks and her family was not consented, because at that time it was not customary to ask permission from the parents also ki their cells are going to be used for the experimental purposes. So in 2013 there was so many issues. There was very big issues by the family of the Henrietta Lacks ki they have got their HeLa cells patent for the for studying all the cancer lines, but still they have not given any royalty or something like that. Anyways, these are different issues, but just remember ki Henrietta Lacks have left the legacy, a remarkable cells. These cells are still all the experimental labs we use these kind of HeLa cells. So let's see what are the different characteristics of these cells. So first is that those kind of cells are they keep on dividing even they reach a particular shape and size and particular density, because they do not require different growth factors. The other is reduced requirement of extracellular growth factors. So in some cases, cancer cells produce growth factors that stimulate their own proliferations. Such abnormal production of a growth factor by a responsive cell leads to continuous auto-stimulation of cell division. That is autocrine growth stimulation. It means that the cancer cells still is producing some substances that are important for their growth stimulation. They do not require extracellular nutrient factors. So in other cases, this reduced requirement of growth factors result from the abnormalities in intracellular signaling systems, such as unregulated activity of growth factor receptors or other proteins, like generally RAS proteins or protein kinases are involved in these kind of proliferations of growth, proliferations of cells, because RAS proteins and growth factor proteins are abnormally regulated, and that is why all those checkpoints are disrupted. That prevents the cell to grow uncontrollably or abnormally. So the other is defective differentiation. The other characteristics is defective differentiation. So what is this defective differentiation? Another general characteristics of the most cancer cells is that they fail to differentiate normally. Okay? What happens ki we have the n number of mosaic cells. We have mixture of cells in which, suppose we have in the female generally 2 X chromosomes are there, and in all the cells 1 X is inactivated. So different, we have the different kind of mosaics. In one cell, this particular X1 is, suppose 2 cells, we can name them X1 and X2. So in X1, this particular cell X1 is inactivated, in this one X2 is inactivated. So we have the mixture of cells, mixture of alleles also. But what happens in the cancer cells, these have only one type of cells. These have the, because they arise from the clonal selection. So once in all the cells only one type of cells is being produced without any differentiation. So in leukemia also they used to produce white blood cells, because otherwise the stem cells can give rise to n number of different kind of blood cells like red blood cells, white blood cells, neutrophils, macrophages, lymphocytes, leukocytes. But in leukemia what happens? All the stem cells are start producing leukemia without any differentiation. They do not have any kind of differentiation. The other one is contact inhibition. Contact inhibition is that when a cell start growing and when it can get contacts with the neighboring cells, cell to cell interaction occurs, and the cell decides or determines ki okay, this is the time I have to stop growing further. So cancer cell actually cancer cells actually do not show any kind of contact inhibition. So they keep on growing even after the neighboring cells are present, and they keep on one layer and 2 layers, they can keep on growing. So what is happening in basically contact inhibitions? So 2 properties of cancer cells affect their interaction with other tissue components and thereby play important roles in invasion and metastasis. First, malignant cells generally secrete proteases that digest extracellular matrix components, allowing the cancer cells to invade adjacent normal tissues. For example, secretions of collagens disrupts all the connective tissues, and thereby the cancer cells start invading connective tissues and enter into the blood vessels. Second, the cancer cells secrete growth factors that promote formation of new blood cells. Okay, suppose cells are growing and growing and growing, they start making the blood capillaries also. So this promote the formation of new vessels. Is the formation of new capillaries, new blood vessels is called angiogenesis. So angiogenesis is needed to support the growth of tumor beyond the size of about 1,000,000 cells, at which point new blood cells are required to supply oxygen and nutrients to the proliferating tumor cells. So such blood vessels are formed in response to growth factors secreted by tumor cells. The formation of such new blood vessels is important in supporting tumor growth and also in metastasis. The actively growing tumor cells are formed in response to angiogenic stimulation, are easily penetrated by the tumor cells because new blood capillaries are already formed, so it will be easier for the cancer cell to invade other tissues also. So that's it. So we know, in conclusion, there are different characteristics. One is they do not show any contact inhibitions. Then they have they do not show any kind of differentiation, and they have what they can show autocrine growth. That means they can produce their own growth factors which acts on the cells and they keep on growing. The other is they are density dependent, they do not show density dependent inhibitions, यानी कि the size of the cells can be increased without any checkpoints. So that's it for the cancer. I hope you have understood the concept. Thank you.